Tonight we will be debating the following resolution. Resolved. Nucleotides can be explained via natural phenomena on the early Earth. Supporting the affirmative will be Lucifer. On the negative, we will have Alexxx. We'll start out with 12 to 15 minute construction speeches, followed by another round of construction speeches, and then some rebuttals. With that, Lucifer, you are up.
Thank you. Okay. All right, so just want to encourage people to go to the link that I provided. You can sort of kind of follow along with my presentation here. And I'm going to dive right in. Okay. So nucleotides formed and accumulated naturally on the early Earth. Specifically, ribose, deoxyribose, and other sugars were synthesized by both the foremost reaction and by amino acid catalysis. Nucleobases were formed from amino acids and formamide synthesis, and these products bonded with each other to form nucleosides and with phosphate to form nucleotides under the conditions of early Earth. To explain this, I begin with a brief description of this time period, an aeon referred to as the Hadean, from 4.6 down to 4 billion years ago. Soon after the formation of the Earth and Moon, these bodies cooled, and carbon dioxide and nitrogen became a dominant part of the Earth's atmosphere. Since photosynthesis is overwhelmingly the primary production pathway for oxygen on Earth, and since it is not detected in significant amounts until approximately 2.5 billion years ago, the planet is determined to have had much less atmospheric oxygen at this time. Evidence from Hadean zircons from the Jack Hills in Australia, dating from approximately 4.4 billion years ago, provide evidence for the existence of an ocean by 200 million years after the Earth's formation. Relevant to nucleobase formation, atmospheric interactions between nitrogen and lightning from 4.4 billion years onward led to the accumulation of nitrate and nitric oxides into the ocean, and also nitrate and nitrite into lakes, ponds, lagoons, and basins after the onset of plate tectonics and the production of continental crust. At the bottom of the ocean and of these other water body types, the interaction of water within or fissures of ultramafic or iron-loving crust results in a process called serpentinization, where iron reacts to yield an alkaline fluid of hydrogen and methane fuel, formic acid, and formate, as well as a suite of minerals and metals, including iron, nickel, copper, magnesium, tungsten, zinc, cobalt, calcium, chromium, and others, that precipitate on the water body floor, comprising hydrothermal chimneys or mounds. Serpentinization has extremely important consequences, and it has been described as the mother engine at the dawn of life. To at least partially understand why, we have to appreciate what are called gradients, or changes of degree that exist within and along the space of a local environment. For example, serpentinization creates a heat gradient, beginning at higher temperatures deep within the fissures of crust, well below the chimney at approximately 200 to 300 degrees Celsius, thereafter cooling to a moderately hot 90 to 120 degrees as it flows and rises and passes through the central channel of the chimney, and then finally down to much lower temperatures of approximately 25 degrees along the periphery of the chimney complex, where the serpentinized or alkaline fluid contacts colder water from the outside. This outside colder water is rich in CO2 from the atmosphere and therefore of acidic pH of a value roughly 5 to 7. There is therefore also a pH gradient between this outer fluid and the inner alkaline fluid of pH 11 to 12. These chimneys are often composed of what are called chemical gardens, interconnected labyrinths of nanoporous minerals, as well as of layered sheets of metal-doped clays and amorphous silica gels. While many different routes for nucleotide synthesis have been explored, my concern will be the processes going on at these sites, or sites very similar in makeup. A nucleotide is composed of three main subunits, or moieties, namely a sugar, a nucleobase, and a phosphate group. In RNA, for example, the sugars D-ribose, more specifically D-ribofuranose, and aldopentose sugar. The nucleobases can be adenine, cytosine, guanine, or uracil. By bringing these three subunits together, you form a nucleotide. From here on out, we explore the sources of each of these components, as well as their bonding interactions, their stabilization, and proliferation. We begin here with sugars. Alexander Butleroff discovered the foremost reaction in 1861, a first reaction I want to highlight that self-replicates or forms more of itself in a process called autocatalysis. The foremost reaction takes place using formaldehyde under alkaline pH in the presence of a variety of divalent metals. It is now well known as a way to make sugars. Formaldehyde itself is a ubiquitous molecule on Earth, and its primary production pathways at vents include the reduction of carbon monoxide and a formic acid, both reduced by hydrogen, as well as minor formaldehyde delivered from the ocean. The diversity of products in this reaction is robust, yielding a variety of pentose and hexose sugars, among a variety of other products in potentially competing reactions, such as the Cannizzaro reaction. The direct yield of important sugars like D-ribose is therefore extremely low, typically less than or equal to 1%. These facts, that competing reactions can take place under these same pH conditions and that the yield is small, have led to skepticism about the relevance of the foremost reaction as a pathway for ribose production for RNA. Moreover, some have argued that a so-called asphalt problem ensues, where products that remain under these conditions often devolve into complex mixtures and tars described as, quote, inauspicious precursors for biochemistry. In evaluating the plausibility of this reaction to yield sufficient concentrations of ribose over time, the following data was discovered from a series of experiments. First, Fiallo et al. Examined the behavior of other sugar products of the foremost reaction that form in much higher yield, such as ribulose. It was found that ribulose readily isomerizes into ribose, showing that the long-term yield of ribose sugars from the messiness of the foremost product is much higher than the direct yield. This study and others point to the fact that aldose and ketose sugars readily interconvert into one another under mildly basic conditions, and in the presence of nucleobases isomerize into ribose and form ribosides, that is, ribose-based nucleosides. They write, quote, ketoses interconvert with their aldose isomers under mildly basic conditions, for example, ribulose with ribose and arabinose. Moreover, most sugars interconvert between their alpha and beta anomers, change ring configurations, and epimerize at chiral centers adjacent to carbonyl groups. These observations offer an alternative and robust solution to the ribose problem, whereby more readily formed ketoses isomerize into aldoses and are trapped in situ by nucleobases to give nucleosides directly, with yields that are not limited by the de novo synthesis of ribose. They more specifically quantify this when they write the yields of nucleosides formed in this reaction reflect the amounts of aldose sugars present when the nucleobase is added to the isomerized mixture. For example, after 360 hours at 65 degrees Celsius, a starting solution of pure ribulose contains 3.3 millimoles of aldoses. The addition of the nuclease to the reaction mixture results in 3 millimole of pentose nucleosides, corresponding to 90% capture of each of the aldoses present. Second, Arthur Omron in 2020 published the plausibility of the foremost reaction at hydrothermal vent environments, in which he demonstrated that flow along the vent's pH gradient entailed that sugars do not remain in the same conditions under which they form. Instead, they flow to the peripheral, cooler, more pH neutral regions within the mound, where they stabilize and accumulate. Omron writes, this study investigates the relevance of the foremost reaction with an emphasis on the membrane-like features of hydrothermal vent structures and their flow-driven, far from equilibrium conditions. Hydrothermal vent chimneys are naturally occurring examples of chemical gardens. Chemical gardens are hollow tubes that form by the rapid precipitation of metal carbonates, phosphates, hydroxides, oxides, and amorphous silica. This work utilized so-called chemical gardens as vent chimney material analogs and shows that calcium-based chemical gardens catalyze the foremost reaction. Additionally, using a simple reaction diffusion model, this study shows that the membrane-like walls of the chemical garden provide a complex environment for the production and accumulation of sugars in the presence of transmembrane gradients. These conditions would indicate the foremost reaction is plausible in the environments of alkaline hydrothermal vents, unquote. Zhao in 2021 showed that ribose preferentially accumulates into metal-doped clays, becoming sequestered from other reaction products, stabilizing within clay layers while retaining its reactivity. Zhao experimentally tested the stability and reactivity of the ribose accumulated in his clays in experiments and found that after 32 days there was no detected degradation of the retained ribose, and that while in clays, ribose retained the capability of bonding with nucleobases to form nucleosides and furthermore readily phosphorylated into nucleosides. Based on these experiments and our knowledge of the Hadean, we can say there's good evidence that during the Hadean aeon, metal-doped clays surrounding hydrothermal mounds on water body floors sequestered and accumulated D-ribose. D-ribose was reactive under these conditions and so would have been capable of bonding to make nucleosides and nucleotides in the presence of nucleobases and phosphate groups. Amino acids are an important pivot in our discussion because they're not only our source for many nucleobases, they also react with metals to produce more ribose. In a paper published in 2005 entitled Prebiotic Carbohydrate Synthesis, it was reported that glycoaldehyde and glyceraldehyde react and the amino acid proline to yield a multitude of sugars, including a 19 to 20% yield of D-ribose. The literature on hydrothermal synthesis of amino acids is itself extensive, and you can see two graphs presented in the link I provided detailing the stock of prebiotic amino acids available to a hydrothermal vent model, including proline, glycine, and aspartate. Glycine and aspartate act as precursors for nucleobases themselves. Just this past year, Stuart Harrison earned his PhD working on nucleotide synthesis under hydrothermal conditions and reports the following, quote, I have identified that the formation of pyrimidine nucleobases is possible in a single prebiotically plausible one-pot reaction system. I have identified that the oxidation of dihydroorotate can take place with multiple oxidants, and copper-2 catalysis can enable production of both orotate and uracil, a nucleobase. Investigating the environmental limitations of this reaction show that it can take place under a wide range of conditions, though optimal conditions appear to be most compatible with an alkaline hydrothermal vent hypothesis. Clay 2022 demonstrated the upstream step of converting glycine to orotate through hydantoin as an intermediate product, and aspartate converts to orotate by adding a carbamol group. Formamide is frequently described as the simplest monocarboxylic acid amide. It is derived from multiple pathways, including heating of ammonium formate, which is itself derived from a reduction of nitrogen and carbon sources. The production of formamide becomes much larger at impact sites, and vents accumulate formamide at the bottom of crater lakes within their porous mineral membranes, as shown by Doreen Neither in 2016. Within 45 to 90 days, formamide concentrations become sufficient for nucleobase synthesis, and sufficient catalysts for the reaction include iron, manganese, and silica oxides. Quoting from Saladino, quote, it has been recently demonstrated that mineral self-assembled structures catalyzing prebiotic chemical reactions may form in natural waters derived from serpentinization, a geological process widespread in the early stages of Earth-like planets. We found that these bilayer membranes made of amorphous silica and metal oxide hydroxide nanocrystals catalyze the condensation of formamide, yielding 4 nucleobases of RNA, 3 amino acids, and several carboxylic acids in a single pot experiment. Besides manganese, iron, and magnesium, 2 abundant elements in the earliest Earth crust that are key in serpentinization reactions, are enough to produce all these biochemical compounds, unquote. Pyrrhotite, a hydrothermal vent mineral, has also been shown to catalyze the formation of uracil at approximately 6% yield using urea, a decomposition product of ammonia, and succinic acid, as shown by Aldecoa in 2013. From here, we can form nucleotides from 2 different pathways. We can add sugars and phosphates together and then add on the nucleobase, or the nucleobase can attach to a sugar, and then the nucleotide is formed with the final addition of a phosphate. Both pathways have been experimentally verified. For example, just as the amino acid proline could react with metals like zinc to manufacture sugars, the amino acid cysteine can react with iron to accelerate sugar phosphate formation, as reported by Pietrafita et al. In 2021. Inho Nam et al. In 2017 demonstrated the formation of sugar phosphates and uridine ribonucleosides using microdroplets. Microdroplet experiments are important because they are mimics of the kind of chemistry you can expect between the sheets of those metal-doped clays, which contain minor amounts of water within their interlayer galleries. This reduced water content is one proven answer to what has been called the water paradox, where water, an essential ingredient for RNA functioning, also favors the degradation or hydrolysis of RNA in bulk water solution. To complete the step to nucleotides, Kim and Benner experimentally demonstrated nucleotide formation from phosphorylated sugars and nucleobases, also in 2017. Following the alternative pathway, Nam published a follow-up piece in 2018 entitled Abiotic Synthesis of Purine and Pyrimidine Ribonucleosides in Aqueous Microdroplets using Magnesium 2+. Takert et al. In 2019 demonstrated a one-pot synthesis of DNA nucleosides using acetaldehyde and water. To complete the step to nucleotides, Ju and Zhang published, quote, aqueous microdroplet-driven abiotic synthesis of ribonucleotides using monopotassium phosphate. They write, quote, here we report that the production of ribonucleotides from ribonucleosides in the presence of monopotassium phosphate, KH2PO4, spontaneously proceeded in aqueous microdroplets under ambient conditions and without using a catalyst. A full set of ribonucleotides, including adenosine monophosphate, guanosine monophosphate, uridine monophosphate, and cytidine monophosphate, were generated on the scale of a few milliseconds. The aqueous microdroplets could transfer the ribonucleotides to oligoribonucleotides and showed mutual compatibility for individual phosphorylation, unquote. Moreover, Rodriguez et al. In 2019 published Confinement and Time Immemorial, where they synthesize AMP, ADP, and ATP using mineral nanopores. Their abstract reads, quote, "We report the successful one-pot synthesis of adenosine mono, di, and triphosphate in the confined space of a mortonite zeolite. This is also the first report of ATP synthesized onto a porous mineral surface. The results revealed a plausible prebiotic route to ribonucleotides and highlighted the contribution of microporous minerals in the origins of life. RNA and DNA require the joining of these nucleotides into polynucleotides. I'm going to close by pointing to 3 articles that demonstrate this final required step to nucleic acids like these. Mizuki published in 2019 that mineral adsorption of nucleotides selects for longer molecules, reaching lengths of up to 100 nucleotides by the end of their experiments. Ron and Mast put the heat gradient to use and found that it increases polymerization by a factor of greater than 10 to the 600, generating polynucleotides over 200 nucleotides long. Thank you.
Well, that was a bit long, but you kind of feel lucky it wasn't. Anyway, so now we will hear from Alex and his constructive. You ready, Alex? It was kind of easy to follow along with the paper you have there. Some of the stuff I understood. Anyway, next. Alex.
I'm here. I'm going to keep it to plain English. Lucy, that was very impressive. So I'm going to speak 2 minutes on myself and Lucy. Lucy knows far more about this stuff than I ever will. There's no question about that. But the real premise of this, and I texted to Pox early, was the question is, how much do we know about abiogenesis? So Lucy gave us a really good model, many good models about what plausibly or possibly could have happened on early Earth, resulting in life, right? My presentation, which is barely my own, I'm not a biologist and I'm less of a chemist. I simply examine and regurgitate what real chemists say, only if it seems to make sense and whether their allegations are supported in the literature. So we'll get to the technical stuff later, but this is what I want to say about origins of life research. Organisms care about life. Molecules don't care about life. Chemistry is utterly indifferent to life, and without a biologically derived entity acting upon them, they never make the move towards life. I'll repeat this once more. Molecules have never been shown to evolve towards life, and any observation of molecules assembling towards life doesn't happen without intervention or a biological entity working on them. What we do see occurring naturally without intervention are molecular assemblies with regular repeated ordering like AAAA or ABAB. These assemblies can be gotten simply because they are thermodynamically favored. We have a chatter who visits the room every day, and she mentions this type of assembly and organization all the time, thinking it can be extrapolated to biologically relevant molecules, polymers, and assemblies. The obvious problem for anyone who thinks that this is the case doesn't understand that life consists of non-ordered assembly. That is, non-regular assemblies of molecules make up every part of us. We have to have non-ordered assemblies to make up the complexity required for living systems. So, where are we in our discovery of how life began? And what does the research look like? Every origin of life research paper, essay that I've ever seen, and the ones that Lucy has presented, like, by the way, Lucy was asked to forward me 4 or 5 or 6 papers so that we can dissect them and see whether the actual experimentation supports the modeling that Lucy paraphrased or put out for us during his first 15 minutes. So every origin of life research paper, essay that I've ever seen is analogous to this protocol. Researchers purchase chemicals of extreme purity to form from a chemical company, often tailored to the needs of the experiment. Researchers mix these chemicals together in sufficiently high concentrations in specific order under some set of carefully devised conditions in the modern lab. Researchers obtain a mixture of compounds that have a resemblance to one or more of the 4 basic classes of chemicals needed for life, or their precursors. So sometimes they'll do an experiment to get to a building block to get to a building block. Those 4 classes being carbohydrates, nucleic acids, amino acids, and lipids. All life we know is composed of these 4 building blocks, and I just went to the wrong page, is composed of these 4 building blocks. So ultimately, researchers are trying to make building blocks or building blocks that are the building blocks to building blocks. So a relay synthesis in order to get to one of those 4 classes. Researchers then publish a paper making a bold assertion about the origin of life from these functionless crude mixtures of stereochemically scrambled intermediates, much like Miller did in 1952. Then we have to deal, number 5, then we have to deal with the ever-gullible reader of headlines who proclaim, see, the scientists know how life was formed. I'm going to take a shot now. One can substitute MDel for either the gullible reader or the researcher in the last sentence. Ha ha ha. I have a feeling tonight we're going to focus on points 1 and 2. The how, where, when, and how much of the starting conditions are prebiotically plausible. Because if they're not prebiotically plausible, but overly orchestrated and contrived, then they're not prebiotically right. And number 2, the contrivance of the scientist. That is, how much of the experiment is carefully orchestrated. In other fields, let's see what we've done in other fields of science. We have satellite connectivity. We have cell phones. We have the structure of DNA, and we can even manipulate DNA. We've identified thousands of functional parts in cells. We've had moon landings, outer space exploration. And now we're planning for Mars. We've had VCRs, then DVDs, then thumb drives, and now we have streaming on any device of our choosing. We've put a computer in almost every home. We've eradicated diseases and treated many more. We've explored the deepest oceans and characterized faraway galaxies. The list of scientific achievements is endless. But it's my assertion that little has changed in the origin of life studies in 70 years. Scientists have conducted thousands of brilliant experiments, but the answers have been few in terms of real answers on any front. But this is such a broad field that the need to pare this scope down to this chat tonight was obvious. So we decided to go focus on the building blocks of life. That was too big. Those are carbohydrates, proteins, and nucleotides. Then we further reduce it to nucleotides and one of their constituent parts, nucleobases. Have resolved the prebiotic pathway to the synthesis of nucleobases is what we've pared it down to. Lucy has provided for me a list of 5 papers that might offer some insight into that pathway. I'm thinking that all of Lucifrase's offerings will be found to be subject to the same implausible starting conditions and implausible contrivance that was mentioned earlier. To his credit, and this is where he's brilliant, he has probably sent me 30 papers in the last 45 days. But again, the scope had to be narrowed. I mean, to take on sugars, lipids, amino acids, and nucleotides in one sitting would be just too much. So we settled on nucleotides. That was his pick, by the way. On the history of these critiques. So these critiques of original life research is nothing new. Prominent researchers such as Karen Smith, Robert Shapiro, Clemens Richer, James Stewart, etc., have all been critical of the plausibility of origins of life research. For your information, those guys are all very esteemed and eminent scientists. Robert Shapiro is dead now, though. To construct any, and this is I'm going to quote Clemens Richer. To construct any convincing theory of abiogenesis, we must take into account the condition of the Earth about 4 billion years ago. We have to use chemical techniques that might have been available on prebiotic Earth. Tides, waves, thermal events, periods of heat, cold, hydration, dehydration, etc. All these things that might happen on early Earth, right? They all may have played a part in this chemical evolution. For a synthesis to be categorized as being prebiotically relevant, it must use chemical reagents, that is the stuff we put in conditions that are presumed to be available on early Earth and accessible on early Earth. For experimental scientists, there is a need to start from defined conditions and results can only be trusted if they are reproducible. So it can't be a one-off experiment. It's got to be something we could reproduce. That's science. So there is little doubt that chemists will work from known quantities of pure chemicals. Such a pure chemical scenario on early Earth is unrealistic prebiotically. Why? because organic compounds produced from simpler starting materials on planets are usually found in mixtures of structurally entangled related molecular entities. Further, the ideal experiment does not involve any human intervention. Action or a reaction network is allowed to unfold, and the sample is only broken up when the experiment has been finished. That's Clemens Richer. He goes on to write, for experiments aimed at demonstrating chemically more complex processes such as multi-step synthesis mimicking biochemical pathways or genetic replication, repeated interventions by the experimentalist, the scientist, have been necessary. Each step needs a specific chemical environment or set of conditions to occur in high yield. For example, the elimination reaction, which is a type of reaction where several atoms, either in pairs or groups, are removed from a molecule, needs different conditions than an addition reaction, any of a class of chemical reactions in which an atom or group of atoms is added to a molecule. And assuming that both will occur at the same time in the same geolocation is unrealistic. That's where the scientist gets involved, and he has to order these things. How much time have I got? Okay, anyways, I'll keep going. Okay. So here's a few issues. Like 4 minutes. Here's a few issues. Molecules have no brain. They don't know where they're going. That's the synthesis problem. They don't know how to stop the course of progression or why to stop. There's no target for the molecules. They don't know when they've arrived at a target, nor do they know how to stop. Let's say we want carbohydrates. You set up an experiment and the reactions are happening. Maybe it's maybe the flask has made a carbohydrate. It doesn't say, heck, we've made a carbohydrate. I think I'll stop the synthesis because I know if I don't, I'll go on and make something else from that. Time, although claimed to be the great savior of abiogenesis, can actually be the enemy. For example, carbohydrates, their kinetic products, they undergo caramelization or the Cannizzaro reaction and they decompose. So in other words, when you're making a carbohydrate, that's not the final product that would form in a reaction. A chemist watches the reaction and they stop it at a point during the progression of the reaction from going on. If left an undirected, unguided reaction, it keeps going towards some other irrelevant garbage. Much of what the Urey Miller flask was tar. It was junk, undecipherable stuff. Carbohydrates are kinetic products, meaning that they caramelize, they polymerize into a bunch of trash just when you take sugar and heat it up on the oven. It turns into caramel. That's what happens to carbohydrates. They don't simply remain carbohydrates. They end up actually breaking down. In the lab, the product is formed at a precise time and the reaction is stopped. When the target has been reached, it is usually stored, frozen, treated with stabilizers and buffers or some other means to prevent further reaction or decay. In the case of our example, a carbohydrate is a desired product. Okay, I'm going to go forward. So what on earth played this role of the chemist? Who stopped the reaction at precisely the right time? Who added additional reagents at exactly the right time? How was the state maintained? Once it reached the target, how did it prevent from ever getting any or decaying into garbage? What else is crucial? A prebiotic system doesn't have the ability to easily purify the structures. You have to be able to purify, because if you cannot purify the byproducts, and that could be in the thousands, build up in the system, and they start using up your starting material, and they start inhibiting the reactions that you want. Chemists will start reactions and purify. That is, eliminate unwanted byproducts. They might introduce a chemical to bind unwanted products, irradiate with UV light, a specific wavelength, alter temperature, alter pH. They might degas a product in a vacuum chamber. There's all manner of ways that they clean up the unwanted, unnecessary, and probably chemical reaction frustrating byproducts. What's doing this on early Earth? How were unwanted byproducts and reaction frustrating side reagents dealt with on early Earth? What else is a problem? Reagent addition is a problem. The chemist constructs the experiment in a way that the target product is achieved. In any kind of multi-step synthesis, certain things are added at certain times. But wait, there's more. Also critical are the parameters for temperature, pressure, solvent, buffers, light, no light, pH, atmospheric gases or no gases, contaminants have to be carefully and crucially controlled. Who's minding all that on early Earth? Later on, I will post excerpts of methodology to highlight the degree of contrivance necessary and accompanying parameters. that are pertinent to Lucy's modeling out, in his presentation. What else? The origin of life. Okay. The backbone of origin of life research, relay synthesis. So the origin of life researcher almost invariably uses relay synthesis over and over again to bypass low yields and purification issues. Read some papers and see how prevalent it is. And it'll be obvious if we dissect the papers. You take high purity A compound and high purity B compound, you get a series of compounds, AB1, AB2, AB3, AB4, AB5, etc. The foremost reaction that Lucy mentioned will yield thousands of products, literally thousands of products. But you want one. Let's say it's AB4. Heck, it's there, right? But researchers don't isolate AB4 from the product of the first step because it's too messy and yields are typically low and concentrations are weak, and it's entangled with thousands of other products. So he says for the second part of the experiment, we're going to use AB4, but you know what he does? He buys high purity, uncontaminated AB4 for the next step in his experiment. Again, don't believe me. Read the methodology. So he'll buy AB4 from a chemical supply house, which is what he does generally, or he may harvest it from a natural source. Okay. Now the claim is AB4 can be synthesized abiotically or prebiotically. So he now mixes AB4 with C, and that yields ABC1, ABC2, ABC3, ABC4, and 1,000 other compounds. Now let's say you need ABC3 for some further synthesis. You don't take it out, right, from the mess that that part of the experiment created. What you do is you buy from a chemical warehouse purified high quality ABC3. Don't believe me, read the methodology. That's how they do it every single time. They can't use the ABC3 because it's contaminated. It's weak concentration. It's entangled in with a bunch of other junk. That's called relay synthesis, and it's a cheat. Read the methodology or supplemental information for any of the papers we might discuss tonight. It'll be apparent. Everyone will show reagents of high quality, isolated and in high concentrations. Oftentimes, use of a compound will be justified by the referencing of another paper. For example, paper ABC might say that formamide was derived in paper XYZ. Read paper XYZ, and perhaps the experimentation is anything but plausibly prebiotic. But because it's been because it was mentioned as a product in that first paper, it's fair game to be used in the second paper. Again, that'll become apparent as we examine some of the papers tonight. Now, I'm just going to put this in a room. This is a list of how much time have we got, by the way? Am I over time? I'm out of time. Shoot. Okay, I'll quit for now.
Okay, that was Alex's statement. I think I got the gist of what he was saying. Anyway, those were, and you know his criticisms, you need to answer them. Next, Lucifer.
Okay. So thank you, Alex, for the opening statement. All right, so there's a bunch of things going on here. So first, he talks about the extreme purity of what scientists will buy for their experiments. This is not a full scope of abiogenesis research. This is how experiments are often done, but it's certainly not how all experiments are done in this field. To buy pure starting materials is necessary in a lot of experiments because you need a control for variables. If you don't know that this is what's causing the reaction to take place, you can't come up with a conclusion to your article. You can't give an account of how life came about because you don't know that it's that particular starting material that's causing the reaction. And so this allows us to establish causation in our experiments. But scientists like David Deamer and many others who actually go out into the field, find natural hot springs to conduct their experiments in, these kind of messy environments that Alex is talking about. And the literature on messy chemistry related to the origin of life is really extensive at this point. Deamer, for example, in his work in hot springs, demonstrated polymerization in this messy environment. How in the world did that happen if it's impossible for this chemistry to be facilitated in these impure kind of scenarios? He described Robert Shapiro as a critic of abiogenesis research, and I think that's kind of unfair to Dr. Shapiro, because he wasn't really a critic of abiogenesis as a field. He was a critic of the RNA world because he was a metabolism firster, which is what I am. He was actually supporting, he was one of the earliest supporters of the kind of view that I'm putting forward tonight. So he also makes this point about bold assertions on the part of researchers, and I really disagree with that, because the vast majority of papers that I read are overly modest in their conclusions. You know, I've seen Dr. Torr talk about this. He discusses it as hype, and a lot of the examples that he gives from the actual literature I wouldn't consider hype at all. He can give examples from like popular media, but that's another thing. Another important thing, the buildup of waste. You know, so like you have a messy environment, but there are certain things that can interfere with your reactions. How is it that a certain portion of the of your product is retained while others are lost? And the way that this works from a hydrothermal vent perspective is that you have adsorption onto mineral surfaces inside of lipid membranes. And so your product is trapped on the mineral surface and contained within a membrane. The processes that go on inside that membrane spit out their product outside the membrane. This is something that's talked about by people like Bruce Lipschitz and David Deamer and a lot of other people working on hydrophobic pockets or lipid membranes and how they work. And so they'll excrete things like water or other products out into the outside environment. Since we're dealing with a vent, this product, this waste product, is taken up by flow through the vent reactor and into the dilute ocean. That waste goes away into the dilute water, but the product is retained inside the chimney structure. That's how you get rid of a lot of waste, and you have a cleaner environment, even though you do still have a lot of stuff going on there. He mentioned the foremost reaction. He says you get all these different products, but you only need one. That's not really true. You don't just need one. You need a lot if we're going to account for the way that life is today. In the pentose phosphate pathway, for example, you have ribulose, you have fructose, you have ribose. So you need those sugars if you're going to account for modern metabolism. You need to account for carboxylic acids, which are the result of that Cannizzaro side reaction that we were talking about. So is that garbage? Is that totally useless or irrelevant to prebiotic chemistry? No, you need that material. Another thing about waste product and about why you end up with just your prebiotic material at the end, also this has to do with thermodynamics. There's this paper called The Thermodynamic Tailwind, and another paper that was referenced to me by this creationist, Rob Stadler. And what these papers talk about is like, you have some basic starting material, so you start off with like 6 different starting materials, and you end up with literally like tens of thousands of products from this. But the vast majority of them, like, and in the paper that I'm referencing from the creationist, literally only 80 to 85 of the products out of these thousands were prebiotically relevant, but they were also exclusively the most stable products. The vast majority of them were far more geared to degrade in comparison to the prebiotic product. This goes to something that Nick Lane and the physicist Victor Stenger talk about. Why is there something rather than nothing? Because something is more stable. The vast majority of your other products are already geared to break down, whereas the products relevant to life are actually far more stable than the alternatives. Let's see where we're at here. Talks about like sequential reactions and the need for human interference. But a lot of the material that I referenced tonight are one-pot experiments or work with what are called continuous reaction networks. And these things involve no human interference. They involve adding a bunch of things into one single pot and watching what happens. So you don't need to have a human come in and add in this step back and then add in this step. Things that do involve human interference, like, for example, changing the pH. Alex talked about the need for variable conditions that he considers unrealistic, but this goes back to the gradients that I was talking about before. You don't just have an environment that has one pH, one temperature, one kind of solution, and that's it. No, this is an environment that has variable conditions in that one environment. You have a pH that goes all the way down from about 5 to 6 all the way up to 12, depending on where you are in that environment. You have a temperature range that starts from 200 to 300 degrees Celsius, goes all the way down to 25 degrees Celsius, up toward the periphery of those vent complexes where your molecules are getting trapped inside those clays, inside those nanopores, and they stabilize there because it's a neutral pH. It's much lower temperature. Talking about how scientists will store and stabilize and freeze. Well, this kind of environment, you have a much lower temperature at the end range where these molecules are getting trapped. They're not at this high temperature where they're degrading rapidly anymore. And so that's how, nature mimics or has its own analogs to what the human scientist does in their experiments. Let's see what else is going on here. Talking about relay synthesis, the way that origin of life researchers work is they examine one thing at a time. To demand that every single experiment start from scratch and go all the way up to, say, a ribozyme is highly unrealistic, even though there are papers out there, like, for example, you can find a graph about the amino acid synthesis that I was talking about before. This is a continuous reaction network, starts with just like ammonium, sulfur, silicate, other base, and ends at peptides covering multiple steps. So those are papers that aren't just relay, but relay is still important in the sense that you're looking at one step along the way and seeing whether or not that step is feasible. And if you establish that, it's certainly not, I think it's an unfair criticism to say, well, that's cheating that you're just looking at that one step. No, if you have, for example, low yield during that period, during that particular step, you can, that's not necessarily a bad thing so long as you have accumulating and stabilizing mechanisms that will allow that low yield to accumulate over time. As long as that's done feasibly, that's A-okay. And I don't consider that cheating at all. And I'm trying to see if there's anything else. Time is the enemy. With this, I'll just close by saying that life works on the scale of nanoseconds. The processes of life are extremely, extremely quick. And at a hydrothermal vent, you're talking about 100,000 years of that hydrothermal vent surviving, being active, and then eventually degrading down, which gives you about 10 to the 24 nanoseconds for this chemistry to start, complexify, and occur, and move to life. And many of the abiogenesis researches that I read, quoting, for example, Lane, that is plenty of time indeed. To close here, I think that, there's been a lot of work done over the course of the last 70 years, and to say that abiogenesis has made no progress is a statement that's in contradiction to the evidence. Like any other field, abiogenesis has open questions, has things that it has not yet solved. But to pretend as if all of this data that I've highlighted here, and that can easily be referenced by looking to Google Scholar, doesn't exist or is insufficient, I think is pushing credulity and is definitely in contradiction to the vast majority of evidence that we found over the course of the last several decades. Thank you. I yield the mic.
That was just under 10 minutes, and I was keeping track. You did answer some of the questions Alex posed. So. Anyway, Alex, your turn.
Yeah. Well, okay. So again, Lucy, you said a lot, right? So the ask for papers was to see whether the papers and the experimentation actually supports the modeling that you're presenting. But you said a couple of things that have me a little bit baffled. You said the products relevant are far more stable than the byproducts that might break down. What doesn't have a half-life? What biologically relevant molecule doesn't have a half-life? It's not stable. Of course it'll break down. Fine, it might land in colder temperatures, but eventually it will break down. It's not thermodynamically, it's not at its thermodynamically most optimum place. It will break down. You said that, and Robert Shapiro, I think, was a metabolism first guy, and he was a critic of Origins of Life research. He earned the moniker Dr. No, because he would go through this stuff and he said, that's not plausible, that's not plausible, that's not plausible. But, and the last thing I'll say is, last thing on that topic I'll say is, I'm going to take my 10 minutes, is I can only work, Lucy, with the papers you gave me, right? So if we're going to talk about sugars, ribose, carbohydrates, anything like that, anything other than nucleotides or specifically nucleobases, I would have to prep for that. I can't respond to that. But in terms of the starting materials, here, this is from one of the papers that you gave me. And this is kind of convenient. D-ribose. That's kind of convenient, D-ribose, right? So he's got D-ribose, was purchased from TCI Laboratories with a 98% purity. That's kind of lucky it was D-ribose, that one enantiomer for the experiment. Anyways, look at the list of materials that are used. They're all high quality, high purity. They're all purchased from chemical supply houses because they need to be. But let's go on. Let's examine methodologies, right? And this is kind of technical, but let's see. This is an example of a guy, one of your experiments, and they're making sugars, right? So forget the technical stuff. I want to highlight some things. A mixture consisting of blah, 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 was added to 3 ml of prepared pentose solution and reacted at 70 degrees Celsius for 30 minutes. The mixture was cooled to room temperature and placed in the vacuum oven to remove ammonia. The mixture was extracted with chloroform for 5 times. Forget that part. Why was that reaction stopped at 30 minutes? Remember, we're making sugars here. Why was it stopped at 30 minutes? You know why, right? But what on early Earth might stop that reaction at 30 minutes? Right? It was stopped at a precise time, and there's a reason it had to be stopped at a precise time. Here's another one. As an example of the contrivance, right? So formaldehyde, 99% purity, good thing for that. Blah, 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 blah. Okay, the presence of formaldehyde did not change the pH of the alkane silicate solution, but accelerate the gelling process. Before the gelling, however, 10 microdrops, 5 milliliters each, and then they tested various metals with it, right? But they had to add that after the formaldehyde was added, but before it started to gel, right? And then the microdrops, those reagents were heated to 80 degrees Celsius for 24 hours. Before that, it wasn't at 80 degrees Celsius. So how does that happen on early Earth? We have this mixing of chemicals, an adding of the metals that you want to test, and then we get a heating, and then after that we get a heating for 24 hours. How do you account for that on early Earth? How does that happen? Right? That's huge contrivance. Now these two are from the same paper. Right? These two are from the same paper. The preparation of reagents for the same synthesis. Right? Here's the first part. Blah 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 blah. No, that's not it. Sorry. Wrong copy and paste. Where are you? Okay, I got it now. Sorry, guys. Shoot. Okay. Oh, okay, yeah, that's fine. That's fine. Okay, these two are from the same paper. That one, right? So we have a solution that's heated to 80 degrees Celsius for 24 hours. From the same paper, in preparation for reagents for the same experimentation, we have this preparation for another reagent, right? Here we go. So that one is heated to 80 degrees Celsius. This one, right, same paper, We need both these for the same for this synthesis. This was added to a flask and it was heated at a reflux for 3 hours. After all the glycine had been consumed, blah blah blah, the H side was added to reaction, flask stirring it over an ice water bath for 5 minutes. The reaction was then heated at reflux for 3.5 hours. The reaction flask was chilled to 4 degrees Celsius for 12 hours. Crystals were then collected using vacuum filtration. This experiment requires the precursors for the final product to be prepared in vastly different ways. Remember, they have to be geographically, geophysically located very, very close, because they're going to be near the same geyser or hydro or vent or whatever it is you want to place this at. But look at the preparation that's required to get those two reagents. How does that happen? That's contrivance to an nth degree. Finally, I guess for now, we have the preparation of equipment and stuff like that. Read some of the methodologies in what might come up tonight. Extreme sanitation and cleansing of flasks, surfaces, drip equipment, test tubes, irradiation with UV light, baking at 500 degrees, washing with millipore. That's water that's first it's been purified, it's distilled water, and then it goes through filtration 10 times. H2O2 washings. Why? Because the presence of any contaminants, including something like magnesium ions, which is everywhere, the Earth's crust is 2.5% magnesium, which is everywhere, might frustrate or thwart the reactions. How is this level of sanitation? Absence of metal ions. How do you account for that on early Earth? More contrivance, right? Okay, and the rest of what I got has to deal with Lucy's paper specifically, which we can go. I got 5 of them. I got rebuttals for the 5 of the papers that he gave me. So I don't know what we want to do next. What do we want to do next here?
We want to do whatever he wants to do and you want to do. As long as you keep on topic. And I think you're keeping on topic. Well, Lucifer, again, the temperature and contrivance argument seems to be, I don't want to get involved in this. Next.
I think we're on our 3rd round, right? Which means rebuttals of 5 minutes, or actually there's 2 rounds of 10-minute rebuttals. So this is our 3rd round of rebuttals, 10 minutes each. And then I think we have 1 wrap-up of 5-minute closing statements. So I'm going to treat this as our 3rd rebuttal. I have 10 minutes and begin. All right, so first about Dr. Shapiro being called Dr. No. Yes, but in the context of the RNA world, that's what he was a critic of. He himself presents extensively a metabolism first scenario. He has it published in his books all the way back to the 1980s. So to say that he's just no to abiogenesis period is a mischaracterization. Shapiro, what he's criticizing is and his criticisms are extensive of the idea of RNA first world. And I actually share his criticisms of a lot of those things. I think that we're dealing with co-evolution of things like RNA, peptides, lipids, and so on. And that's why I detailed a lot of this chemistry, showing you that this environment, which has a great deal of diversity located in that environment, is able to facilitate a diverse range of chemistry, allowing for co-evolution of these different things. So like, talking about some of the excerpts that you provided of like before gelling, he adds something and so on and so forth, but you're actually talking about 2 different things there, because at the beginning of the paper, what he's doing is in the portion that you quoted, he's synthesizing the mineral membranes first. So that is what that's for, and he's mimicking it, like the metals are there first and so on and so forth, because he's building the membranes. So he has to mimic the process of how you do that geologically. How does geochemistry actually make these mineral membranes? And that's what he's doing there. Then from there, he's going on to do this chemistry involving the condensation of formamide to nucleobases. So when you're talking about well, he needed to do this, but of course he needed to do that if he was going to synthesize those mineral membranes. That's how you do it. That's how nature does it as well. And then why stop at 30 minutes? But you don't have to stop at 30 minutes. Like the way that these reactions work at a hydrothermal vent, the synthesis is steady, it's ongoing. Every single second, the same reactions are taking place because you have this flow going on both sides of an interface. As long as the flow on both sides are steady, which it is for 100,000 years, you get continual synthesis of this product, which is why low yields build up over time. But you say eventually it will break down. Yes, it's true. Eventually it does break down. And there's two things going on here. The first thing is that if your products break down before you can synthesize more net product, then you have a problem. But if your rate of synthesis is steady and your rate of breakdown is relatively slow, you have a net gain in product, which is exactly how this works. It accumulates over time. You need to have some kind of degradation taking place because that's also how life does it. Life doesn't just have anabolic metabolism of building things up. It also has catabolic metabolism of breaking things down. So the fact that things will eventually, along the time scale, decay says very little about whether or not the model is overall plausible. What you need to check is whether or not synthesis is steady to the point where you get net product over your degradation. And the kind of model that I'm putting forward here, and I want to really emphasize this too, because talking about when I say model, right, it sounds as if like, well, this is kind of speculation. But I want to emphasize how it is not simply speculation because we know that these processes were taking place. So I quoted at the beginning of my presentation the nature of the Hadean environment. We know certain things about the Hadean environment because we have Hadean zircons that survived that we can look at to see what the environment was like at this time. We also have, like for example, you take a look at this paper, Evidence of Early Life at Hydrothermal Vents by Dodd, published in 2017. The hydrothermal vent that he finds is dated at 3.7 to 0.2 billion years old. So we know that there were hydrothermal vents going, occurring at that time. We know from the zircons that there were oceans at that time. So we know that these things were there, and we can study the chemistry that goes on given the conditions that were given. And we also can look at modern day analogs, like for example, Lost City or Stryton or the Prony hydrothermal vent at New Caledonia. And we can study those things to see how the chemistry works there. And all that we need to do when we do that is to recognize the differences between those vents that exist today versus how they were during the Hadean. Take those factors into account and we can come up with something that is most probably true, or at the very least has a high degree of verisimilitude, very close to what the truth is. But you don't have to stop at 30 minutes and this whole thing about tar formation, stuff like that, because again, your products don't stay in that environment where you're forming your product. The formed product flows to another area toward the periphery of the vent, in a cooler area, in a more pH neutral area where it doesn't form tar or degrade or anything like that. Instead, it stabilizes within metal doped clays, within nanoporous minerals at low temperature, at neutral pH. And when you have a situation like that, you're not degrading rapidly, and yet at the same time, you have a steady synthesis accumulating over time. Let me see here. That's also, emphasizing here again, different temperatures. How did you do that? Because you have different temperatures in the same environment. Recognizing cracks, you have to 300 degrees Celsius as it flows up. Once it goes out to the periphery of the vent, it goes all the way down to 25 degrees as it interacts with the ocean water or the pond water or the lake water or whatever. And so you have variable conditions in the same environment, and the product goes through those different environments through flow, and that's how it works, and that's exactly how it mimics the way that humans, in terms of like adding in a step here or changing the pH there, they're actually mimicking how a variable environment like this actually works. And you don't need any human interference at the vent itself because it's flow that takes care of that for you. Mg2 plus, and actually there, he's actually what you're referencing with Dr. Tour. He's referencing the cleaning of various product, and he thought that Mg2 plus was completely cleaned off the product, but he's actually dealing with basaltic glass, and that innately contains Mg2 plus. So Dr. Tour is actually mistaken there. He thought that you're washing away the Mg2 plus, but the mineral itself is composed of Mg2 plus. You're not washing that away. That's actually contained in the chemistry. This is another problem with critics like Dr. Tour, is that while he's a respectable synthetic, he knows very, very little about geology, and it's very important to understand how geology, geochemistry, and geophysics actually work in these environments in order to understand what you're really doing. When you're washing things off to do away with contaminants, that's again a necessary part of experimental protocol. If you do not do that, you don't know that the reaction is innately the result of what it is that you're using in your experiments, because it could have come from somewhere else. This is another reason why, like, working with formaldehyde, for example, can be really difficult, or working under anoxic conditions can be really difficult, because oxygen can break in. Formaldehyde is a ubiquitous contaminant. It's very difficult sometimes because of how easy external formaldehyde can make it into your experiment. You have to control for those things. And if you don't, then you don't actually have a conclusion. You're not demonstrating causality. The fact that they use products is what allows us to know that it's that which causes the reaction. And if you want to make sure that it can still take place in a messy environment, then you introduce messy components, which is what researchers have done time and time again. I gave the example of David Deamer before, not a lab, but going out into the natural world to test in the messiest of environments to see whether or not he could facilitate things like polymerization. A last one that I'll reference here is by Benner in 2017. He specifically wanted to address Alexx's problem about using like pure materials or synthetic materials. And so he went all around Canada and the Americas and so on and collected natural minerals to see whether or not they could adsorb and concentrate RNA. And his results, using only the natural minerals, pyrite, for example, natural mineral found at hydrothermal vents, 90% effective adsorption. Magnetite, another one found at hydrothermal vents, 30% adsorption, and they work just as well as your synthetic components. So you can test the natural samples as well to see if they work, and they have, and they do. Yield the mic.
Well, okay. Okay, Alex, you got 5 minutes to come back at what he said. To the extent you want to, you can do whatever you want.
Well, again, you're referencing material that I'm not privy to, right? I can't speak to some of the the introduction of messy materials in their experimentation. You didn't send me that. What you did send me, though, everyone, because I looked at supplemental information, everyone pristine from the chemical warehouse, 95% purity or higher, sometimes 99% purity, undiluted, and that was their root stock. That was their stock for their synthesis. Now, if you, and we should do this another night, if you want to forward me papers in which they use something less than those pristine materials and pristine conditions, then that would be interesting too. But I like to respond to the stuff that you sent me 5 papers. I like to respond to them. I guess I'll do that. So I'll give you links, titles, and then problems. So this is the first paper you gave me. Its title is this. Its title is that. And part of the methodology is this. And remember, they're trying to create nucleobases, right? So see that pyrite column was heated at 100 degrees Celsius and flowed just to maintain a constant pressure? Okay, that's fine. I mean, if that's what's going on, that's what's going on. But then let's examine the stability of RNA bases. right? Let's examine the stability of RNA bases. Come on, link. Here's a link. There's a link. Let's read it. Right. So the assumption you have to make on your side is that the flow is the savior for this synthesis in a deep sea vent or something like that, right? Because as you read here, high temperature origin of life theories required that the components of the first genetic material are stable. We therefore have measured the half-life for the decomposition of the nucleobases. They've been found to be short on geological timescale. At 100 degrees Celsius, your synthesis is at 120. But at 100 degrees Celsius, the growth temperatures of the hydrothermophiles, the half-lives are too short to allow for the adequate accumulation of these compounds. Half-life for adenine, why didn't that take? I cut and paste that improperly. Half-life for adenine, I'll give it to you later. For guanine, it's a year. For uracil, 12 years. For cytosine, 19 days. And they continue to write. They write, therefore, unless the origin of life took place extremely rapidly, in less than 100 years, high temperature origin of life may be possible, but it cannot involve adenine, uracil, guanine, or cytosine, which are the nucleobases we're talking about. The same experiment that creates the nucleobases required for nucleotides, those same conditions destroy this stuff in the blink of an eye, geologically speaking. That's a description that the paper gives it, not me. So isn't that a problem, Lucy? That's the first paper you gave me. I don't know if you want to comment on that one. Go ahead.
Well, she's a minute left, but it's okay. Lucy, here we go again.
Okay, so I think we're on our last 5-minute statements, yes? And so I'm trying to pull up the stability of the RNA bases paper. I've read this one before. And so, you're saying that I'm assuming that flow is going to save this synthesis, but it's incorrect to say that I'm assuming, because what I have sent you in terms of papers is how the flow actually works and accumulates products at the periphery of these vent complexes. So you can look, for example, at the paper that I sent you by Zostak and Budin in 2009 using a thermal gradient, where you have a hot source and then outside colder area. And what this does is it creates a force and flow called thermophoresis away from the heat source toward the colder regions. Budin and Zostak in 2009 showed that you could accumulate both lipids and nucleotides in the colder areas in these micropores like 1,000-fold, like ridiculous numbers of accumulation. Dieter Brown and Mast have shown the same thing. There's a paper, 2013, Escalation of Polymerization in a Thermal Gradient, where they talk about exponential accumulation of nucleotides, even down to nucleotide monomers. But because the flow it is determined by weight, so it actually selects for the longer particles, those are the ones that are most likely out of all to get stuck in the colder regions. The monomers, however, can also get stuck there. And so you can look at many, many different papers that use a heat gradient and flow to see what happens to your prebiotic product. And time again, what they find through things like there are different kinds of transport mechanisms. They have long names like diffusiophoresis, electrophoresis, thermophoresis, and they work by flow, electrical interaction, heat gradients, gravity, and so on. And these have been shown time and again to accumulate prebiotic products that's originally synthesized in a hot area, like that pyrrhotite at 120 degrees, but does not stay there, but instead flows to the periphery of the vent and gets trapped in those nanopores or intercalated into those clay layers at much, much lower temperatures of like 25 degrees. And in the Stability of RNA Bases paper, I'm pretty sure, I'm pulling this from memory because it's loading up really slow on my laptop, but the most unstable one is cytosine. And at 0 degrees Celsius, it has a half-life of 17,000 years at 0 degrees Celsius. So you could see that at very high temperatures, it's a blink of an eye, it degrades. But if you have a system with flow involved, it does not stay at those higher temperatures. It goes to the colder areas, at which point it becomes exponentially more stable. And so I think that this is a major problem in terms of the creationist critique of abiogenesis, is that there's this presumption that your environment is totally static. It does not have dynamic variable conditions in the same one environment. If you say pH 11 to 12, it has to stay pH 11 to 12. If you say temperature at 120 degrees Celsius, you're only working with 120 degrees Celsius. But that's not actually how the vast majority of models current in Origin of Life actually work. Not just at the vent environment, but also with like the nuclear geyser model that gets brought up here sometimes, also with the tidal pools on land, where you have a hot day and a cold night. You have wet conditions when your tide is up, when your water content for your pond or your lake is up, and then during evaporation it goes down, and now you're under dry conditions. In the same environment, you have variable conditions. And so this kind of critique that relies on just these one conditions as if it stays static is geologically unrealistic and not actually how it works. It's based on a false assumption. And with that, I don't think I have much more to add at this point, so I will yield the mic. Thank you.
Okay. So you guys can go back and forth for 5 minutes as long as you want. Yeah. I don't think you have to. I think we've answered the problem. We've solved the problem here. We've answered the question. Alex?
Well, I had 4 more papers to go through in terms of what I wanted to say about them. I mean, he sent me 5. I thought since he took the time, I would address all of them. Okay, here's another one. Okay. We have a single. First of all, this one is from. This is a synthesis from formaldehyde. Formamidine. Formamide. Sorry, formamide. This is a synthesis from formamide. I mean, there's a heat problem there, right? Because we get the synthesis at 80 degrees Celsius, and we know what the half-life is at temperature already. But here's the other problem with this one, right? So we read this. Formamide seems to be key in so many of these experiments. I mean, I didn't just look at yours, but I looked at other ones, and formamide seems to be such a key. Formamide pools of furthermore, pools of formamide on early Earth would likely have found it difficult to form spontaneously. That's one paper. The link is there, so you can examine all. Now, here's one that really should bother you regarding formamide. Further, not that one. No, hold on. Formamide formation requires reaction between HCl and water, and the latter continues to convert formamide into ammonium formate. Recently, it has been shown that the phosphorylation of glycerol increases remarkable in the presence of formamide as a solvent. However, all these cases assume the presence of pools of formamide, which may be unlikely as formamide hydrolyzes to ammonium formate at relatively low temperatures when water is abundant. Remember, your model takes place underwater. Right? So unless that water is somehow eliminated, and you mentioned microdroplets, and maybe that's a solution, but formamide, in the presence of water, is going to hydrolyze into ammonium formate, right? So that'll disappear quite quickly if underwater. But there's more. It's not the only paper. Formamide, because it's so key. For example, formamide has been used as a solvent for the phosphorylation of nucleosides such as adenosine and uridine. Despite these successes, it has been argued there are few geochemical routes to pure formamide puddles, and every one of these experiments, by the way, uses neat formamide. Neat formamide, and you know what that means. Puddles on the early earth silicate promoted. Okay. So this paper doubts we had formamide synthesis, right, where you can get puddles of it accumulating. But who knows? Maybe you got something to the contrary. And one more. One more regarding formamide. There's a large amount of literature showing that on heating neat pure, that's a symbol for formamide, above 100 degrees Celsius, amino acids and nucleic bases are produced. However, neat formamide is not likely to be geochemically plausible. There are no known feasible processes for the generation of neat formamide in a realistic, primitive, early Earth environment. The hygroscopic nature of neat formamide promotes HT hydrolysis, indicating that unless there was a robust process of continuously generating neat formamide, pure formamide, it would not have been abundant on early Earth. So some of the papers rely heavily on the availability of formamide, but I just gave you 3 links that suggest it likely wasn't that available on early Earth. Of course, in a lab, we can do anything, right? We set it up to work. We set ourselves up for success. And they have formamide, neat formamide, which means it's pure formamide to conduct their experimentation. Realistically, though, is that available on early Earth? A lot of literature says no way. Yield the mic.
Well, if you want to go, keep on going. I ain't going anyplace.
Okay. All right. So there are 2 main things that were brought up here. One is on the plausibility of forming formamide, and the other is that the synthesis that I'm talking about takes place, and so you have this threat of hydrolysis. So first, I want to concentrate on formamide formation. I think it's interesting that Alex is making this, because it was actually Alex who sent me, I think, one of the most important papers on this topic that talks about the manufacturing of formamide at impact crater lakes. So you have a meteorite that comes in, hits, and it creates some kind of lake system. And then what we're talking about here are called impact-induced vent systems. These are continentally based vents on land that form under lakes or ponds and so on. And these lakes and ponds are formed meteorites. The paper sent to me by Alex specifically discussing the plausibility of accumulating formamide from at impact crater lakes is exponentially higher. That's the first point. And so that paper pointed out the geochemical plausibility of formamide forming in those environments. Second, I referenced earlier Doreen Neither's paper at 2016. And what she did was she concentrated on the idea of, even if you started off with like very minor amounts of hydrogen cyanide, which I don't even use that at all, because you get ammonium formate at vents from a totally separate route. And if you heat ammonium formate, you get formamide. But starting from either synthetic route, starting from hydrogen cyanide or directly from ammonium formate, you accumulate formamide in hydrothermal pores, and she calculated 45 to 90 days based on the fact that formamide is thermophobic. So it too acts in just the same way as our prebiotic molecules, where that heat gradient drives it away and accumulates it into those colder areas of the vents. And her calculations are between 45 and 90 days before you have enough formamide concentration in the micropores in order to conduct this kind of chemistry. And so I think those are 2 really important papers that talk about the geochemical plausibility of this synthetic pathway based on the fact that meteorites at creating these impact lakes exponentially increases formamide synthesis. And then the fact that the vent structure at the bottom of the water body concentrates the formamide that's produced very, very heavily. And within 45 to 90 days, again, you have enough to proceed. With respect to water hydrolysis, this is taking place in a vent environment. Yes, it's underwater, but it's not in bulk solution, which is really important. It changes the whole deal. When you're talking about water, very, very minor amounts of water entrained in nanopores or in between clay layers, it's not this bulk solution where you have only water, nothing but water, and it's just water all day. Instead, you have very reduced quantities of water, which reduces the rate of hydrolysis. And in turn, because you're concentrating these prebiotic molecules in this area, the concentration flips the thermodynamics. It goes from favoring hydrolysis to favoring polymerization. This is something that Kunin talks about. He has this paper called RNA Reactor for the Origin of Life, talking about the previous work that I cited in Zostak 2009, working with that thermal gradient and concentrating nucleotides and lipids. And what he pointed out is that when you have that kind of concentration, you're no longer favoring hydrolysis. And likewise, I want to emphasize again, you are not in bulk solution. You're in a confined space. And so I want to talk about this portion by emphasizing two things. The first is about messy environments. I really encourage everybody to just go to Google Scholar and type in messy and prebiotic messy environments, messy chemistry, and see the countless articles that come up of experiments done under these kinds of conditions where critics like Dr. Tour will say that it's impossible for anything to form in these kinds of environments. See the countless papers operating on these with these kinds of conditions to see how they actually form, and also look at people like Deemer who actually work in natural environments with success. And then the last thing is look at prebiotic nano confinement and look at the difference of how chemistry works under confinement versus in bulk solution, and how the rate of hydrolysis is totally, utterly different. Hydrolysis is favored in bulk solution because you have nothing but water, but you have controlled water or structural water in confined spaces or in clay layers, and that totally changes the game. Now you're not favoring hydrolysis. Now you're favoring complexification. Yield the mic.
Okay, I have a suggestion. You and we could do this again. I want to do it again 30 days from now. You've heard what my objections are, or what, not mine, they're not mine, obviously, gosh, I'm not a chemist, they're not my objections, but you heard what some of the objections are. Maybe you could send some papers that address those ones. But there was one more paper that, because you took the time, right, might as well cover it. That's not it. What's wrong with my cut and paste, man? Hold on a second. Cut. That one. This might even be your favorite one. Plausible prebiotic one-pot synthesis of orotate and pyruvate suggestive of common proto-metabolic pathways. Okay, so we want orotate, right? We want orotate. How do we get orotate? A reaction between two prebiotically plausible building blocks, hydantoin and glyoxylate, generates both nuclear-based orotate, precursor of biological pyrimidines, and pyruvate, a core metabolic in the citric acid cycle, amino acid cycle, blah, 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 blah. Okay, basically, we want orotates from, to summarize, copy, we want orotates from hydantoin and glyoxylate. Right, okay. But that second one is a problem. So we're going to synthesize that from that. Glycoxylate from glycine and formaldehyde. Great. So let's look up that. Let's see how let's see how we can manage that. And here you go. Paper. Study. Another paper. Plausible prebiotic origin of a glycosate non-enzymatic transition reacting glycine with formaldehyde, published in journal Synlight. Okay. So this isn't one you sent me. This is my own stuff. So here's some material for you. right? So just to summarize, we want orotate, but we got to get orotate from hydantoin and glyoxylate. And glyoxylate we got to get from glycine and formaldehyde. Well, there is a way of synthesizing that. Let's look at it. And here it is. And there it is. And let's see what we get. So there's the experimentation. In the supplementary material, the yields varied under varying temps and pHs and time. They range from 88 parts per million to 171 parts per million. That's an example of relay synthesis, right? There's no way you can get this to work in a lab, right? That is to go from here up to orotate using a concentration of 171 parts per million. A parts per concentration would never, you'll never find it in any experimentation anywhere, is my bet, right? That would never be a starting reagent, right? Here, but let's look at what they used. Let's look at what they used, right, in the next order of synthesis. So remember that, 171 parts per million. What did they use? Glycolic acid solution, 50% weight, right? So you get a liter of this stuff by weight. 50% of it is glyoxylic acid. How many orders of magnitude is that greater than 171 parts per million? Using this, you might get an experiment or a reaction to run. Using 171 parts per million, I doubt it, but maybe you can present something to the contrary. So in order to get orotate, which would work, we have to synthesize a couple of things to get to orotate. And it doesn't look as though the yields of the previous steps would cut it in terms of a real reaction. Maybe you got some comments on that.
Pox, I was kind of holding back to see whether or not you wanted to say anything. I'm going to treat this, I think we're at, are we at conclusions? Do we want to go one more rebuttal round? I'm kind of open. Based on the format, I think we're at conclusions. If you guys want to go more rebuttal round, we can. Okay, so the first thing is this complaint about relay synthesis that you have. It's pretty peculiar to people like Dr. Tour. And I talked about this before, that if you start with, if you are just examining one step, I don't consider that cheating. What you have to do is you have to perform a calculation to see whether or not, if your one step involves low yields, which I think is your complaint here about parts per million, if your synthesis results in low yields, you need to consider whether or not your model, the way that your system actually works, does it accumulate low yields over time? And does it deposit your products in an area where they can stabilize such that your net degradation is lower than your overall rate of accumulation and synthesis? And so that's an important thing to consider if we're talking about stepwise examinations of abiotic processes. Another thing is that I pointed out in my opening statement 2 routes to orotate. So glycine to hydantoin and glyoxylate is one, but the other one is aspartate. And what you do is you add a carbamol group to aspartate, which also turns into dihydroorotate and orotate. And what Harrison showed with the PhD thesis that I focused on before is that if you have copper-2, you not only do you form dihydroorotate and orotate, but you also form uracil, a nucleobase. So there's multiple prebiotic routes from amino acids to nucleobases, and given the conditions of the environment, what happens is that your product is deposited in what are effectively incubators, like these clay layers, at low temperature in areas where they can survive for extended periods of time. If we're talking about how long these things will last under these conditions, and like how long molecules can last overall, like something say the other day, Alex, you quoted this paper about RNA degradation, and you gave this reference for like 2 to 3 minutes. But an important thing there, and I wanted to touch on this, is that you were referencing something that was talking about RNA degradation within the cell. But RNA degradation in the cell is controlled, just like RNA synthesis is controlled. It's deliberately broken down by the cell. Doesn't answer the question of how long these biomolecules will stay around and potentially accumulate in an environment. And so, you can look to even our studies nowadays about like ancient DNA that's been discovered. The oldest one that I know of was contained in permafrost in mammoth teeth over 1 million years old, which is quite a bit longer than 2 to 3 minutes. The environment that I'm stressing tonight does not involve permafrost, but it is at a much lower temperature of 25 degrees Celsius. That's where all of the final product ends up concentrating, and that's where your further complexification takes place. 25 degrees Celsius is room temperature, so you're not operating under these extremely hot conditions where time and time again, I agree with Stanley Miller, these products will break down extremely rapidly if you're at these higher temperatures. But once more, to stress one last time, if you're in this environment, room temperature, incubated within clay layers or mineral nanopores, that is not the case. They last for a much longer period of time. The paper that you referenced from Miller cited that at 0 degrees Celsius, cytosine has a half-life of over 10,000 years. So, at 25 degrees Celsius, it'll be a little less than 10,000 years, but still on the order of thousands of years. And yet you have a steady synthesis at the same time, accumulating it constantly. The paper that I referenced from Zhao 2021, accumulating ribose in metal-doped clays, he left his product, his ribose, he watched it accumulate in metal-doped clays. None of the other sugars concentrated there, but ribose specifically did because of electrophilic interaction. It was adsorbed onto and then intercalated into the clays. He left it for over a month, came back, and tested to see whether or not that ribose was reactive, and he formed with that ribose nucleosides and then nucleotides with that ribose after leaving it for over a month. And when you think of, and there was no detected degradation at that time, and think about the fact that you have over 30 days of steady synthesis behind you of more ribose, and yet the product that you synthesized at the very beginning is still there and still reactive. That shows accumulation over time. So even if you do start out with a low yield, as long as you have these kinds of conditions, that's a way of circumventing that problem. If you have low yield in a hot environment, yes, that's implausible. It's going to break down very quickly. Your rates of degradation are going to outpace your rates of synthesis. But when you have much more mild conditions, it's not the case. And many experiments have shown that you get accumulation rather than degradation on net. Yield Mike.
All right, so just examining one of the models that you mentioned. So what's her name with the 45 to 90 day accumulation of formamide in microtubules, right? Did I get that right? 45 to 90 days? You get adequate accumulation of, okay, concentration, okay. So think of it, Lucy. So this formamide is accumulating in these microtubules, right? And it's safe and sound. So how does it get to nucleobases? Heat, right? Suddenly, has to be suddenly, there's an application of heat, right, to synthesize the nucleobases. And then, after that accumulation has happened, this heat has been applied for a certain period of time, that product is whisked away to another portion of this deep sea vent. But it was okay to hang out in these microtubules for 45 days, not being synthesized, right, not affected by heat, because obviously concentrations weren't high enough for anything to happen, for reactions to happen. Or maybe it was, right? Maybe it was hot enough from day one. But then that heat would have for sure destroyed the cytosine, maybe not the other ones, but for sure the cytosine. So how does this work? Is it first in, first out? It gets in there, synthesized, whisked away, and then it starts to reaccumulate for 45 or 90 days? How does that work? Yield the mic.
Okay, with that one, look at the link that I posted in the chat. You guys will see this image of polymerization by a thermal gradient. And the way that this works, if you're synthesizing at the central channel with the hot alkaline fluid, what this does is there's at first convective flow. So you're synthesizing products and it's going back and forth between a hot and a cold source. And during this initial time period with this oscillation, various reactions take place. What Dieter, Brown, and Mast show that during the phase you get exponential increase of polymerization. So those nucleobases that you synthesize, they start to oscillate between hot and cold at first, and they start to undergo their reactions. Then as they gain in weight, because you're synthesizing heavier product, they flow out toward those colder areas and accumulate there into pockets. With the pocket, there's a hotter area to the pocket and a colder area to the pocket, which you see in the image that's displayed. And they convective flow within the pockets. And so there's a cold area and a hot area to the pocket. Because the hot area has thermophoretic behavior, and what that means is that it's kind of like picture if you guys have ever seen like a heat rod, and if you're ever like smoking a cigarette, or if you're smoking a joint or something like that, and what happens to the smoke as it approaches that heat rod, you'll notice that the smoke veers away from the hot rod, and that's called thermophoresis. The heat actually creates a force that pushes it away. So within these pockets, what they do is they spend the vast majority of their time in the colder regions of the pocket. But with that convective flow, they do pass through a hotter portion. But because it creates that force, their length of time that they stay there is much, much shorter than the length of time that they spend in the cold pocket. Some don't even convect after a while. They just sit there and accumulate in the colder region, whereas others continue to convective flow around the hotter portion and continue to do stuff. In the colder regions, though, like for example, if you want to add a phosphate group to form a nucleotide, that's favored at colder temperatures rather than hot ones. This is something that's talked about by Lane and a bunch of other people, that it's colder temperatures that are more prone to phosphorylation reactions. And so, it's not like they just get to the cold area and then don't do anything anymore. No, that's actually where, if you want to really look at where complexity and lifelike processes start to develop, it's in those colder areas. But your initial synthetic route starts at very hot temperatures and then goes out to the colder areas. So hopefully that explains how it works. Yield mic.
Thought Pox was going to win. Well, yeah, it kind of explains how it works as a model. So you mentioned a couple of links, but I didn't have the opportunity to examine those, right? I took great care to address the ones that you did send me because you took the time. Can you send me the ones that support some of these other, this other modeling that you just mentioned? And I'll throw it out there. If you want to do this again in 30 days or something, I'm all for it. So I have nothing else to say right now. Oh, just one more thing. Yeah. Okay. I mentioned you. So you mentioned another pathway for orotate, and that's fine, because the one that I was reading about, I got too much material here. I'll be quick. Sorry for the delay. Oh, here it is. Okay. The one I was reading about was about glycine, right, being rootstock for orotate. So prebiotic synthesis of glycine here. If you're going to get it from that route. Prebiotic synthesis of glycine from ethanolamine in simulated alkaline hydrogen event. So here's the problem I had with ethan. I'm just going to call it ethan, right? Because I don't use these words a whole lot. Ethan. where did it come from? All I could find, all right, I mean, we synthesize it today, no problem, but in terms of natural occurrence, all I could find was ethylamine arriving perhaps from meteorites, where there's, you know, stellar, outer stellar chemistry where we detect ethylamine, sure, but how did it get to the geolocation where it can be used in some of the modeling we've talked about tonight. So, but you derived orotate from another synthesis, so maybe this is a moot point. Just saying. That was a dead end.
Okay, so Fischer-Tropsch synthesis is something that I wish I could have talked about tonight, but didn't really have the time. It's also relevant to nucleobase formation. It's been identified as the formative route for nucleobases and meteorites, and it also takes place at hydrothermal vents. Basically, this works by using carbon monoxide and a bunch of different metals. And what you produce in these reactions are a bunch of different hydrocarbons. So you produce things like ethane, propane, and a bunch of like lipids and fatty acids and stuff like that, as well as minor concentrations like nucleobases and so on. And so you get your ethanol from serpentinization on the one hand, Fischer-Tropsch on the other, both facilitating these kinds of hydrocarbons. And then on the other hand, your amines, which are your nitrogen source, those come from reduction of nitrate and nitrite from the outer ocean or the outer pond or lake water as that comes into your clay layers or into your vent matrix. This reacts with things like green rust or with komatiitic rocks to reduce nitrate or nitrite to amines, to ammonium or ammonia, and that's where you get your source. And so if you're looking for how you're making ethanol amine, it's those chemical processes coming together. You have your ethanol from the serpentinization Fischer-Tropsch on the one hand, and you have your amines from your nitrate-nitrite reduction on the other. Yeah, yield mic.
Okay, with this I'll be done for sure, and I don't know if anybody wants. I don't know if there's gonna be a question answer thing. So with this I'll be done. So you sent me 5 papers. That one was only an abstract. You sent me 30, but you and I agreed that you gave me your favorite 5. Of those 5, when you read the supplemental information, all of them used purchased from the chemical warehouse, high quality, high concentration reagents. Okay, only one involved a little bit of what's the word I'm looking for? When you weaken the concentration, oh my God, dilution. Only one of them involved or included any dilution in them. Okay. 4 of the 5 had multi-step processes where the way you read it, and you say it's just mimicking what's happening in nature, and perhaps that's true, but I don't know that to be true. 4 of the 5 of them were multi-step synthesis where you stopped the reaction, absolutely stopped the reaction, usually the ones going on at high heat after a certain period of time, and then had to treat that yield with a buffering or a degassing or a whatever, right? So my point is, from the 5 that you sent me, it would be hard to say, yeah, this is prebiotically plausible. I mean, just the rootstock materials being so pure and not, quote, messy, I don't know, would make one skeptical. Well, not just me. It makes world-class scientists skeptical. And you mentioned Tour, and it's not Tour that said the human. I posted a blurb from the other fella. It wasn't Tour that had a problem with the methodology. I mean, not him alone. You guys are all hung up on Tour in this room because he's the one that's making noise right now. So I guess I'll leave it at that.
All right, thank you, Alex, very much. I've enjoyed this conversation. I'm going to address your last points here and consider it my closing statement. If Pox wants to take questions from people, I'm happy to do that. Basically, I'll leave it up to Pox and Alex about audience Q&A. All right, so like the first thing about using the pure starting materials, that's something that I think I've addressed several times during this debate about it being important to have those kinds of experiments because it establishes causality, but that abiogenesis research is not limited to these kinds of experiments, and there are many different experiments that have been conducted using natural minerals, natural starting materials, or just outright working out in nature to see whether or not these kinds of experiments work. And I'd be happy to send more papers to you, Alex, about this kind of work that's been done. And also, once again, to really encourage people to just go to Google Scholar and put in prebiotic messy chemistry or prebiotic messy environments and see the countless papers that are popping up. There's this other thing called interference chemistry, where you're still working in a lab, you're not working outside in nature. But what they do is they change so many parameters to the experiment to see just how robust the conditions can be for the chemical situation to work out well. And so like these are many different ways in which abiogenesis research works to make sure that some kind of proposed pathway is in fact prebiotically plausible. A thing that I want to mention in passing, talking about working with pure formamide, there's a paper from Saladino, who's the main guy who's been pushing formamide synthesis over the years, and he has a paper explicitly working with formamide water mixtures, not just like pure formamide. And you can actually, there's a really, this is still something that I think needs to be worked on, but there's a lot of work working with RNA DNA transition, because if we're saying that RNA was kind of doing a lot of the grunt work at first and then DNA came later, which is questionable now because a paper that I referenced earlier was a one-pot synthesis of DNA nucleosides. And again, to emphasize, one-pot experiments and continuous reaction network experiments are totally hands-off. But regardless, like there's a lot of work that's being done showing the differences of behavior and stability between RNA, DNA, and RNA DNA chimeras in different mixtures of formamide and water. And I don't want, there's a lot to talk about with that, but I don't really want to get into it. I don't really have the time at the moment. I'm trying to think if there's any last things to wrap up with. I apologize, I'm kind of losing my thought here as a wrap up. But so I think I'm just going to close at this point by saying that I'm open to having another conversation with you, Alex, at some point. I'll be honest with you, there's probably not going to be just 30 days from now because I was looking forward to getting on with Polaterality and Magellan and other people about some things that they wanted to have me study and talk about. But in the future, I am open to doing another one of these. And I want to thank you very much for participating with me. I had fun. So talking about human intervention, this is what I'll close with, which is, so again, you have to ask yourself, does your model include a change that if a human makes a change in the experiment, is that something that has an analog in the physical environment? I agree with you, Alex, when you say that Tor is not the only one pushing this idea. There are many researchers who complain and whine about human intervention in abiogenic scenarios, and I actually really try to explicitly craft a lot of what I said not to use human intervention as much as possible. But if there are experiments that use human intervention and yet the physical system that you're working with involves those changes, then there's no cheating going on. If you have, for example, you want to study what happens when some product from a lake meets an ocean, you first create your ocean mixture, and then what does the human do? He interferes by adding in the lake solution. Is that prebiotically unplausible? Does that never happen on? No, of course. The lakes travel into ocean water. When the human person, involves himself there, he's studying what is happening when you do that. And nature works that way all the time, especially in these environments that have a dynamism about them, variable conditions within the same local environment. So with that, I'm going to close, and just to thank you guys again, and I appreciate it. Yield the mic.